Hello guys, welcome to Dr. Science. So in my previous video I talked about the overview of acylglycerol biosynthesis, and many of the students were having doubts regarding how glycerol 3 phosphate or the triose phosphate is formed, because from triose phosphate itself, if we do a series of acylations, we can eventually form the product which is your phosphatidate, and from phosphatidate we can usually synthesize your diacylglycerols, from which we can synthesize your triacylglycerols, choline or phosphatidylethanolamine, and and the other thing is your cardiolipin and your phosphatidyl inositol, which are all of the components present in your phospholipids. Now, how this triose phosphate is formed, we have to understand this, because many of the people were having doubts regarding it. So, what is a triose phosphate? Triose phosphate is nothing but your glycerol 3 phosphate. Now, I already told you in my previous video that glycerol is a 3 carbon structure, and to the third carbon we have a phosphate group, hence it is called as your glycerol 3 phosphate. Now, this glycerol 3 phosphate is usually formed via two pathways. One is your glycolysis pathway, and the other one is your triacylglycerol breakdown pathway, or we can simply say that there is lipolysis of your triacylglycerol. First we will talk about the glycolysis pathway, and then we will talk about the triacylglycerol pathway or the lipolysis pathway. So, as you already know that in glycolysis, we will first talk about the glycolysis pathway. So, as you already know that in glycolysis pathway, usually the glucose will get eventually converted into two components. One is your glycerol 3 phosphate, and the other one is your dihydroacetone phosphate, and this dihydroacetone phosphate can be converted into glycerol 3 phosphate by an enzyme called as your glycerol 3 phosphate dehydrogenase. So in my previous video I explained this one clearly, so I'm not going to explain it here. Okay, so as you can see here, from this glycerol 3 phosphate eventually we can form the eventually we can synthesize our product, which is your phosphatidate, by a series of acylations. So this is what so as you can see here, this is the triose phosphate which was formed in the glycolysis pathway. Now, if you are clear with this, next we will talk about the lipolysis pathway. I want everyone to concentrate here. Lipolysis pathway. So in lipolysis pathway, as I already told you, that we have the glycerol. This is the glycerol, and to this glycerol we have 1, 2, 3 fatty acids, and this entire component is your triacylglycerols. So there is an enzyme called as your lipase. Now, what does this lipase will do? The lipase will do hydrolysis of this triacylglycerols, and whenever there is hydrolysis of triacylglycerols, it will break down into two components. One is your glycerol and the other one is your fatty acid. As I already told in my previous video, the fatty acid will enter the circulation, and from the circulation it will bind with the albumin and will reach the target tissues. Now this glycerol will get converted into glycerol 3-phosphate. Okay, now what is the enzyme responsible here? The enzyme responsible here is a kinase. Usually kinases have a phosphate present on them. Usually the function of a kinase enzyme is to put a phosphate on the glycerol. As I already told you, glycerol is a 3-carbon structure, 1 2 3. So eventually this glycerol, on the third position, there will be a phosphate added. What is the enzyme responsible here? The enzyme responsible is your glycerol 3-phosphate kinase. Okay, so glycerol I'm sorry, here the enzyme is glycerol kinase. Okay, so glycerol kinase will do phosphorylation of glycerol and convert the glycerol into glycerol 3-phosphate, and this is what is happening in the lipolysis pathway or the lipolysis pathway. Okay, as in the case of glycolysis pathway, I already told you that dihydroacetone phosphate can be converted into glycerol 3-phosphate by the enzyme glycerol 3-phosphate dehydrogenase. So these two were the main pathways through which we can synthesize the glycerol 3-phosphate, and eventually this glycerol 3-phosphate can be converted into phosphatidate. So glycerol 3-phosphate, what was it? It is a triose phosphate. Okay, so that's how a triose phosphate such as your glycerol 3-phosphate can be synthesized from your glycolysis or your lipolysis pathway. Thank you guys.